My name is John Anderson. I'm the co-chair of this symposium along with Jody. I come from Lund University in Sweden. Please don't leave too fast at 9:30 because we have some important announcements to do with buses leaving and things like that. At this time, I'd like to introduce our speaker, Brendan Frey. He received his PhD from the University of Toronto in 1997. Immediately after that, he was a Beckman Fellow at the University of Illinois, and thereafter professor at Waterloo University, that's near Toronto in Canada, and then since 2001, he's been a professor at University of Toronto. He has cross appointments in computer science and the Center for Cellular and Biomedical Research at Toronto. He's been the author of some 100 papers and one book called Graphical Models for Machine Learning and Digital Communication, and even at quite a young age has received a number of awards within the University of Toronto and within Canada for his work. Many of us first knew him for his work at factor graphs and turbo coding. He was right on the ground floor, or the 0th floor, I think, as we count in Europe, with that. But since then, he's gone on to work in areas related to the genome of mammals, actually. I don't think you're up to people yet, as far as I know. Brendan was concerned 15 minutes ago when he came in that he forgot the obligatory slide filled with abstruse mathematics. But he's made up for it in other ways. He's involved in some controversies, including in science. That's what we really like in plenary lectures, people involved in science controversies. And that will come into some of his work that he'll present here today. Last, and I think perhaps least, he wanted me to point out that he's visited Lund, Sweden. And in fact, it was an interesting occasion in 1996 at the very first turbo coding symposium that was held in that year. And I remember that, and that was his almost first paper in turbo coding. So here he is, Brendan Frey.
So, can people hear me? Good. Thank you, John. So here's the title of my talk, and I'm going to get right down to it and talk about the purpose of my talk. So the idea here is just it's pragmatic. I want to identify aspects of molecular biology that you could potentially make contributions in, okay? So I'm not going to address theoretical issues. I really want to be pragmatic here. So let's come back to the title of the talk. I want to get an idea of how many people know what the genome is. So raise your hand if you know what the genome is, roughly speaking. Well, that's terrific. Okay. So what are the first 20 letters of the genome? What I'm going to talk about is actually beyond the genome. So I will do a brief introduction to the genome. But what I'll be talking about is actually what's called the cellular transcriptome. Okay? And the cellular transcriptome in many ways is much more important than the genome because the cellular transcriptome contains the information for actually performing functions in the cell. I'll tell you about the cellular transcriptome. I also want to emphasize areas where you can actually see, when you peer into the biology, you can see codes and signals. So there will be hopefully some connections that are made to things you already know about. And then finally, I do want to tell you about my personal voyage into this area. 10 years ago, if you'd asked me, are you going to work in biology? I would have burst out laughing. I would have felt a bit insulted. I hadn't even thought that biology was a real science. However, I think biology is finally becoming a real science. So I want to tell you about that personal voyage because, again, maybe there will be some connections to your interests, and if you're interested in working in this area, some issues that you might have to deal with. All right, so first of all, the genome. Just a brief review for those of you who aren't familiar with it. And I'm used to using my laser pointer here, and there's only one screen. There's only one screen that I can point at, of course. It's a laser pointer. So you won't be seeing any green dots on this screen, and I apologize for that. If you want to track the green dot, you'll have to look at the other screen, this one over here. So the starting point, and this is something that I think should appeal to all of you here, the starting point for molecular biology is a discrete symbol sequence. So the symbols are G, C, A, and T. These are called bases. And I'll use in my talk red with an underline to indicate a definition. So whenever you see red with an underline, try to remember that term. I think there are 10 terms that I introduced in this talk. All right, so examples of biological sequences: genes, DNA, chromosomes, proteins, HIV, viruses. So lots of these things are comprised essentially of discrete symbol sequences. Terrific. That looks good from the point of view of a person who works on coding or information theory. The starting point when you look at a cell and ask, how does the cell make use of this discrete symbol sequence to convey information, is chromosomes. The chromosome consists of DNA, which is a discrete symbol sequence, and there are two kinds of processes that lead to replication or inheritance of this chromosomal information. The first one is cell replication, where a cell with a set of chromosomes divides in two, giving each of the daughter cells a set of the same chromosomes. And the other one is sexual cell reproduction, where two cells with different chromosomes combine, producing a new cell with a mixed set of chromosomes. But both of these techniques are essentially ways of propagating chromosomes. And I'm not going to talk about this today, how these mechanisms work. We're going to be more interested in what happens afterward. So the genome, so here's a definition. So for the purposes of our talk today, the genome is just the chromosomal DNA sequence from an organism or species. Okay? So Jody O'Sullivan's genome, that's a genome. We also talk about the human genome. What's the human genome? Well, it turns out if you compare the genomes from two people in this room, they'll be nearly identical. Okay? Nearly identical. So that means when we talk about the genome of a species, we're really talking about the typical genome for that species. Here are some examples of genomes. The human genome, 3 billion bases, so that's how long it is. It's just one long sequence. It's broken up into chromosomes, but you can just think of it as one long sequence, 3 billion bases. Interestingly, that's about 750 megabytes. You can actually load it into your computer nowadays. So there's an interesting development in terms of complexity of the computing devices. The mouse genome is actually quite similar, 2.6 billion. And that's why, if you just look at these crude numbers here, the length of the genome, you can see that mouse and human are a similar length. And of course, they're very similar in terms of many of the processes, such as cell walls, different kinds of cells, hair cells, brain cells. The fly is actually quite similar in length, 100 million bases. And even yeast is a respectable 13 million bases. Okay, so that's just a quick overview of the genome. And the last topic I'll mention in the context of the genome is something you've heard about, a gene. What is a gene? Well, again, for the purpose of our talk today, it's just going to be a subsequence of the genome. It may be contiguous or non-contiguous. It's just a subsequence of the genome that codes for a functioning biomolecule. So a functioning biomolecule, we'll think of that as just some particle that gets produced, from the genome and goes on to perform some function. So maybe the particle is involved in building the cell wall, or maybe the particle is involved in regulating other genes in the genome. So it's just a biomolecule that has some function. So the gene is a subsequence of the genome. That means if you go into the genome and fiddle with that subsequence, that functional biomolecule won't work the way it should have. All right, so that's how we'll define a gene. So what do we know about genes right now? Well, there are libraries of genes. You can take a look at those libraries and how they map to the genome. And interestingly, known genes currently only map to 1% of the genome sequence, so a very small portion of the genome sequence. The other interesting aspect is this library is very much in flux, always changing, and that pertains to the controversy that John mentioned before. I'll come back to that in about 20 minutes. And it's probably far from complete. So even the definition of what constitutes a gene is changing. So this definition I gave you here is not the traditional definition of what a gene is. Okay, so to understand a little bit more about genes and what they do, we'll now talk about the transcriptome. And as I said to you before, this is what I think is more important than the genome. This is the part of the set of biomolecules that actually performs a function. So the genome is our digital backbone of molecular biology. It's this nice discrete symbol sequence. And in the spirit of the World Cup, the transcript, or the transcriptome, is just a set of biomolecules that emerges from the genome. So different parts of the genome get activated, leading to these functional biomolecules, these transcripts. Okay? And these are really the things that perform functions in the cells. So let's take a look at the traditional gene. This is what people referred to as a gene 10 years ago. Traditional gene, we have our DNA, and essentially what happens in the DNA is a piece of it gets read off. There's a machine working along the DNA through a process called transcription, producing a transcript, which is chemically formed from RNA. This RNA is then translated into a protein, and the protein can then go off into the cell and perform various functions. Let's take a closer look at this process here for transcription. First of all, the information is encoded in the chromosomes. These are tightly packed, so they have to be unwound to expose the DNA. The DNA strands then separate, as shown here. A set of proteins works its way into the separated strand of DNA, reads the DNA, and produces this output transcript. Now, something interesting here, if you were to take the entire DNA in one of your cells, just one of your cells, and unravel it to the point that it needs to be unraveled here for transcription, then the DNA in one of your cells would be about 3 meters long. Okay? So you can see that those chromosomes are really, really tightly packed. What about this other process, translation? So translation takes our transcript that we just produced and makes a protein. So the way translation works is we take the transcript, and a set of proteins, again, works its way along this transcript, brings in other components, and forms the protein sequence. So really, it's just this nice abstract notion of a discrete symbol sequence, like a ticker tape, and a machine working its way along the ticker tape, copying out components. So there are 3 kinds of molecules I've shown here. The first one is the genome, which we've talked about. The second one is the transcriptome, which I'll be talking about for the rest of today's lecture. And then the final output is a set of proteins, which are called a proteome, which I won't be talking about today. So let's take a closer look at transcription. I want to identify some of the codes and signals involved in transcription. So this is what, if you zoom in on a sequence of DNA, a subsequence of DNA that forms a gene, this is what you'll see. You'll see components that are called exons, which are things that will actually end up in the final protein, things that are called introns, which are sequences of the DNA that do not end up in the protein. And then there's an upstream region in the sequence that's involved for regulating the production of the transcript. All right, so these 3 different components I'm showing you here, the exons, which end up in the protein, the introns, which don't end up in the protein, and the upstream region of the gene. All right, so this is the DNA. It's been exposed, right? The strands have separated. The sequence has been exposed. What happens is regulatory proteins, if they're present in the cell, bind to the upstream region of the gene. And I'll talk about that in just a moment, how that works. They bind to the upstream region of the gene, and once they've done that, they then bind or recruit transcription proteins. So these are proteins that are going to do this transcription process. So the transcription process, the transcription proteins bind to these regulatory proteins, okay? And then this machine just works its way along the DNA and produces the output transcript. Okay, it's as simple as that. So let's take a look at the upstream region, codes and signals. What do we see? Well, the way a regulatory protein binds in this upstream region is that there's a sequence that's specific to that protein. So here I'm showing an example of a sequence that actually is known to be involved in regulating transcription. So if a particular gene contains a sequence, a code word, like this, and if there's a regulatory protein present in a particular cell that matches that code word, it will bind and activate transcription. Now, if the protein is present, but the corresponding code word isn't present in the gene won't get transcribed. If a code word is present in the gene, but its corresponding regulatory protein isn't present, the gene will get transcribed. Okay? So these are the 2 components that need to be present, the code word and the corresponding regulatory protein, in order for transcription to begin. So the code in this case is a set of regulatory code words. The signals, the things we can measure and tell us about the code words, are the concentrations, the abundances, the amounts of the regulatory proteins, these proteins that regulate transcription, and also the output transcript. So for example, if you look at a large number of samples and you find that a particular gene, you get a lot of that transcript when a particular regulatory protein is present, that's evidence that there's some code word in that upstream region that helps transcription occur. All right. Now, don't think too hard about this analogy, codes and signals. Obviously, in biology, you can't control the code word. So from an engineering perspective, you'd like to place your code words far apart in some space. In the case of biology, we don't have much control over that. You might say that nature has hopefully placed them far apart so that they're distinct, but nature has done what it's done. We're subject to the rules of nature. We can't design the code. All right, so it's just a loose connection in a sense, but I think it sheds light on what the underlying problem is. So how about the transcript? Once this transcript has been produced, how is it going to produce the protein? Well, there's a step called translation I told you about, but translation doesn't occur right away. First of all, the introns have to be removed. So this transcript, which contains the introns and the exons, has to be spliced, just like you'd splice together a tape. I guess you don't splice together CDs, but anyway, in the old days, there were these things called tape players that had magnetic ribbons. And you'd actually splice the ribbons together in order to remove pieces of sound sequence that you didn't want in the final output. So that's how splicing occurs. Basically, you have a similar situation as what we saw before. A set of regulatory proteins binds to, say, the intron region. Okay. These regulatory proteins will then recruit splicing proteins. The splicing proteins then essentially just splice out the intron. Okay, and now those two exons have been stuck together, the intron's been removed. So that's the step that occurs before translation. The translation process would now work its way along the sequence, and it's only seeing exon sequence. It'll produce the corresponding elements that are needed in the protein. However, this is something that's really only become evident in the last 10 years, is there's a huge amount of genomic diversity introduced by different kinds of splicing. So we can take the same original transcript and splice it in different ways. So let me show you an example of that. So again, we have some regulatory proteins. In this case, we have some that bind to the exon and another one that binds over here in this intron region. Okay, so again, this protein recruits the splicing proteins. They work their way along, but in this particular example, the protein that's bound to this exon blocks the splicing from occurring. So it just knocks away the molecule. The molecule then identifies this regulatory protein over here that it can latch to, and the splicing occurs differently. Instead of splicing the first exon to the second one, the first exon is spliced to the last exon. Okay, now this will create a different protein sequence when it's translated, and it'll in fact carry out a different function. And so it turns out in the last 10 years, we found that a lot of diversity is introduced by this process. Again, let's take a closer look at the manner in which splicing is regulated. If we take a look at the sequence, here's the intron, here's the exon. There are short code words bind to regulatory proteins, just like in transcription. The properties are quite different. For example, these code words tend to be much shorter than the code words that are used to regulate transcription. And also, it turns out that in splicing, there are a lot more of these code words. So really, if you like, the code consists of combinations of short code words. And the combinatorial ways in which these short code words can be put together in order to produce the overall regulatory effect. So a very interesting set of rules that we could then investigate and hope to detect that really correspond to the kinds of ideas from codes and signals. All right, so I've told you about a couple specific mechanisms, transcription and splicing, both of which have these components of codes and signals. I'm going to return to a higher level view of what's going on, and what I want to talk about now is the modern transcriptome. So before, when I was talking about transcripts, I was talking about these genes that lead to proteins. Well, the modern transcriptome looks a lot different. So the modern transcriptome, again, we have our genome and our sequence in the genome. Transcription would take a subsequence of this DNA, produce the transcript. All right, looks like that. Again, the exons are shown as boxes, the introns are shown as lines. Splicing would now remove the introns and produce a transcript that does not have introns in it. So when I show these little red upside down V's, those are indicating a splicing operation, the removal of an intron. All right, that leads to a protein through translation. The protein can now go back into the nucleus or wander around the cell and perform a function. However, as I just told you, this other process allows the same gene, the same initial segment of DNA, to lead to different transcripts. So let's take a look at an example here. Suppose this subsequence of DNA, this gene, is transcribed in both brain and liver. Okay? So I'm showing you really two different cells, one from brain, one from liver. but just to keep the picture simple, I'm just going to label them brain and liver. All right, well, this one gene could be spliced in different ways. In brain, perhaps all of the exons are included, leading to this transcript, leading to protein A, whereas in liver, perhaps this exon is excluded, not included in the final transcript, and that'll lead to a different protein that performs a different function. Okay, so really the library of transcripts compared to the library of genes is potentially exponentially larger. Exponential in terms of this complexity of splicing. If there are 100 exons in a transcript, you can imagine the number of potential transcripts that could be produced. Sorry, if there are 100 exons in a gene, you could imagine the number of potential transcripts that could be produced. Now, the last component I'll talk about in the modern transcriptome is transcripts that are actually not traditional at all, in the sense that they don't make proteins. Okay, so here's a piece of the genome that gets transcribed, but it doesn't go on to make a protein. This RNA molecule floats around in the nucleus or goes out into the body of the cell and actually performs a function. So these kinds of biomolecules have been known about for a long time, but recently the importance of these biomolecules has become increasingly recognized. Finally, we have non-functional transcripts. So this is the set of molecules that make up the modern transcriptome. And it turns out to be very surprising in a number of ways. So let me just tell you about a few of the surprising current results. Right? This is all very leading-edge research, results that have come out in the last 3 to 5 years. So these results could change. Our current view, the modern transcriptome, well, first of all, the number of genes. Turns out that in terms of these genes that make proteins, okay, it turns out in terms of those genes, the number of genes in the human is somewhere in between the number of genes in yeast and some plants. Okay? So we have about 4 times as many, according to the current view, we have about 4 times as many genes as yeast, and we have about 1/4 as many genes as some plants. Another interesting fact about the genome, I told you that the set of known genes comprises 1%, only 1% of the genome sequence, a very small fraction. But a recent study showed that over 1/2 of the genome is transcribed. So this transcription machinery is actually transcribing 1/2 of the genome. So what's all that other stuff doing? What's that other 49% of the genome doing? It's either non-functional. There is known aberrant, what's called aberrant transcription, where the machinery just accidentally locks onto the DNA and transcribes. But there could be a lot of molecules that we don't know what they do, and they are functional. They're not traditional. They don't encode proteins. They don't make proteins, but they have functions. I mentioned alternative splicing. Another surprising result that's emerged in the last 3 years is that it looks like over 60% of human genes undergo alternative splicing. So this mechanism is not something that just happens once in a while. It seems to be prolific, especially in complex species. So for example, in yeast, there's hardly any alternative splicing. I think there's 1 or 2 known cases. In humans, there's tens of thousands of known cases. So over 60% of human genes are thought to undergo alternative splicing. And in terms of this diversity, I mentioned there's this potential for exponential growth in the number of these functioning transcripts as compared to the number of genes. And it's not just potential. There's a gene in the fly, the common fly, that's spliced in 18,000 different ways. One gene, 18,000 different ways. 18,000 different proteins. Okay, so this mechanism, and I think it should appeal to this audience because it's combinatorics, right? It's really about the combinations of putting these exons together. Now, are these important? Well, over 20% of human diseases have been associated with deviations in these regulatory code words that control alternative splicing. Okay, so things like cancer is known to be associated with errors in these code words that control splicing. So it's important functionally as well. What other surprises are there? Well, I mentioned these non-traditional transcripts. So these are things that have really only received attention in the last 3 or 4 years. There were known examples before, but a fairly small number. But these days, we're detecting about 10,000 new non-coding transcripts, these new non-traditional types of transcripts each year. So there's papers regularly appearing in the journals such as Science that are reporting large numbers of these new transcripts. Okay, so that's the transcriptome. And I hope I've been able to convince you that it's pretty interesting. and there are potentially some interesting research problems there for you. So I want to talk, not too long, but just briefly about the resources. How would you actually approach a problem if you decided to work in this area? Well, there's two kinds of resources. There's the human resources and also the databases that you can just download. Well, human resources, you get a collaborator, a biology collaborator, and what can that collaborator do for you? Well, first of all, sequencing. So that means if you actually have a notion, you've used some computational technique to predict a transcript, then you can go to your collaborator and say, I think this is what the transcript is, and there's a way your collaborator can essentially create a hook, like a fishing hook, corresponding to that predicted transcript. You can then just dip it into a tissue sample and see if anything latches on. Okay, so you can find out if there is a sequence. That's called sequencing. Another technique that your collaborator could help you with is called microarrays. And a microarray allows you to say, well, I'm not really sure exactly what I'm looking for, but I have a whole bunch of predictions for short subsequences of transcripts, say 60 bases long. Okay? And I'd just like to get an overall view of what's going on in the genome, what's going on in terms of the activation of all these different kinds of transcripts, whether any of those transcripts correspond to my set of predictions. And so microarrays allow you to do this en masse. So it's a very high throughput, large scale kind of assay. I'll talk about that more in a moment. And another one is mass spectrometry. So if you are interested in working your way up to the genome, making predictions about proteins, there are techniques now where you can take an extract from a cell and fetch out the proteins and then try to identify the actual sequences corresponding to those proteins. Now this technology is very crude right now. So it's a bit hit and miss in terms of whether it'll work. But in principle, probably in 10 years you'll be able to do what I just said with mass spectrometry. So what about databases? Another approach is try not to get too involved with this experimental work and try to just download a database. So you can download the genomes. If you go into Google and type genome, you'll very quickly find a repository where you can download a genome. Human genome was 750 megabytes. It's just a sequence of GCAT. It's that simple. This is important, genome annotations. If you'd like to know what are the current genes, what are the known genes, and where are they in the genome, well, you can just download these genome annotations. They'll tell you where the exons start and stop, where the gene begins and ends, things like that. There are also libraries of observed transcript fragments. So over the last 40 years, as people have done research in molecular biology, they've, for various reasons, fetched out short sequences of transcripts and then actually sequenced them. They found out what the sequence is and they put it in a library. These are called express sequence tags. But essentially, they're just short sequences that have been observed. So you could ask the question, do those short sequences correspond to certain regions of the genome? How do they correspond? Can you find correlations between these short sequences, these short transcript sequences? If you can find correlations, maybe you can say something about the existence of genes or transcripts. And then also microarray data sets. So instead of designing your own custom microarray experiment and collaborating with somebody, you can also just download a microarray data set that tells you, for example, how different genes or different transcripts are activated in different tissue samples from a mouse or from yeast or from human. So I said before that I'm going to tell you about a personal voyage. So my personal voyage involves microarrays, so let me just go through this technology in a little bit of detail. So the idea of a microarray is, again, you'd like to ask the question, is this short transcript present in this particular tissue sample? And you'd like to do it en masse. You'd like to ask that question for hundreds of thousands of short transcripts. So the way a microarray works is you take a 1-inch by 2-inch biology slide, you attach to it a whole bunch of probes. Each probe is a sequence of a DNA sequence or an RNA sequence, so GCAT, and you design that probe to be complementary to the sequence you're interested in. You'll see why in a moment that it has to be complementary. All right, so you've designed your microarray. You then take your tissue sample, your cell, extract the transcript. There's techniques for doing that. You then put a fluorescent tag on that transcript. So some molecules that will fluoresce when you hit them with a laser, and you'll see why we do that in a moment. And then you just wash your sample over the microarray, and whenever the transcript sees a complementary sequence, a complementary probe, it'll hybridize. So remember, G, C go together. G sticks to C, and A sticks to T. Okay? So the transcript will hybridize to a probe if it's present. Then you can wash the remaining sample away and scan it with a laser. And if you see a bright red spot, that means that the transcript that's complementary to your probe was present in the sample. All right, so that's the way this technology works. Now, these probes I've shown here, I've shown 5 on the slide. Of course, there are many more. You'd have up to 100,000 probes. Not only that, but you don't just have a single molecule stuck to the slide in each position. You'd actually have sort of a forest of molecules. And not only that, they're not going to be only 5 bases long, which you can imagine is not very specific. If you just look at the length of the genome, right? There are going to be a lot of sequences just at random that will stick to these kinds of probes. So the probes will be longer, 30, 50 bases long. Okay, now how do you actually make the slide? And this really isn't important technically for today's talk, but it's fun. So I thought I'd show you this. One technique is the inkjet printer technology, which my collaborator is a co-author on. It appeared in Nature Biotechnology in 2001. You literally just take an inkjet printer, and instead of red, green, blue, and black, right, the 4 color bins that are in your inkjet printer at home, you just put G, C, A, and T. Okay? And then what you do is you print the probes on the slide layer by layer. Okay? So if a particular probe is GGAA, then your first printing, you'll print a G there. The second printing, you'll print a G. Third printing, A. Fourth printing, A. So to come up with probes of length 60, you'll just do 60 printings. And the picture that you're printing is just the different spots. So if you have 100,000 probes on the slide, you'll have 100,000 of these little spots of G, C, A, and T. Okay, well, microarrays have been around for about, 7 years or so, useful microarrays. And the first microarrays in the late 90s were very noisy. That was the main problem with these things. And I'm sure those of you who know about microarrays, you think noisy, very noisy. They would have about 5,000 to 10,000 probes, but modern microarrays are quite different. So modern microarrays, sub-gene resolution, so you can really get a lot of information about the genome, what's being activated in the genome. Over 200,000 probes per slide now, so you can really probe for a lot of transcripts, not just 5,000 to 10,000. Also, they're low noise, so the noise levels have really come down. And the original microarrays, these ones up here, you typically have multiple probes for each transcript because of this noise problem. The modern ones, only 1 probe is needed. And also, they, of course, come down in price. And so you can use multi-chip design. So if you'd actually like to, say, probe for 1 million transcripts, then you can design 5 slides. It doesn't cost too much. It's about $700 per slide right now. All right. So now my personal experience, my personal voyage, and the main reason I'm telling you about this is because I want to make a connection to things you're familiar with and how they could be used in molecular biology. So a lot of what I've talked about so far has been really molecular biology, and I'm not an expert on molecular biology, and most of you probably aren't. So what I'd like to do is make the connection to things that you're familiar with and make a concrete connection as well, show you how you could make a contribution. So this is based on a collaboration with Tim Hughes, and he's a microarray expert, so he designed the microarrays for this research. By the way, it's really great collaborating with biologists. They like the principal investigator biologist likes to be the last author on the paper, and we like to be the first author. So it's a match made in heaven. Okay, so this work appeared last September in Nature Genetics. And the idea was trying to find out, exhaustively search through the genome and find out the library of transcripts, the transcriptome. Okay, so let's just ask this question. What is the gene library? Well, there's been a lot of controversy in this area, actually for a long time. Part of the controversy stems from the observation that it appears that the human has about the quarter of the number of genes that's in a plant. We like to think of ourselves as being more than plants, so a lot of this is just egocentric controversy. But there's also more recent controversy. So that kind of controversy has actually mostly been settled. But there's more recent controversy, and that's what I'll focus on today. Okay, so it's good to start with an insult, right? So the Phantom RIKEN Consortium is a group of about 200 researchers who are focusing on building a library of the transcriptome. That's one of their main goals. And this is a comment that appeared in a recent issue of Science in March 2006. And this is sort of the center point of the controversy. Let me tell you a bit about how we got to this point, and then I'll tell you how it got resolved. So research on the transcriptome, and this is a modern view, of course, consists of two approaches. One is the genome approach, where you take the genome sequence and then you look for these code words I told you about, these code words that tell you where the transcripts are, where the exon is likely to be spliced, things like that. And you say, well, given the known set of code words, where are all the regions of the genome that could be encoding, could end up in transcripts? Okay, so that's one approach. It's genomic based, it's really just computational. You look for transcripts by looking for patterns in the genome. And of course, that only started after the genome was sequenced in 2001. The other approach is detection of transcripts, and this is a very old approach, but only in modern times, around 2001, did it start to become a high throughput approach, where you could actually take tissue samples and then ask the question, what transcripts are present in these tissue samples? So our project started in around 2003, and as I just mentioned, finished up last year with this publication in Nature Genetics. And we actually combined the two approaches. So we fused these two sources of information. We were looking for code words, but we were also looking at tissue samples and asking the question, do we actually see the transcripts in the tissue samples? It makes sense to use both sources of information. All right, here's a modern, well, I should say a recent view of what's going on in terms of the library of undiscovered genes. So if we look at 2001 and say, what was the library of known genes, and ask the question, what exists beyond that library of known genes? Here are the estimates. So in 2001, the first draft of the human genome was completed. The estimate was that there were about 10,000 new genes. The modern estimate in 2004 was about 3,000. If you look at the transcript-based approach, that's the stuff shown in red, okay? In 2001 to 2003, the prediction was that there was about 300,000 undiscovered transcripts. And more recently, 2004, a paper by Bertoni et al. In Science estimated that there were roughly 11,000 new transcripts. Okay, so that was the picture leading up to 2005. So now let me tell you about our project. So in our project, we have microarrays, we have tissue samples. Let me tell you about the microarrays first. We took the genome, we looked for code words that would indicate potential places in the genome where transcripts could emerge, okay? And we actually looked very far in terms of the distance from those code words. So we tried to drag in a lot of portions of the genome. And the result is that we identified 1 million possible exons. Okay, so this is just a computational technique, so I'm going to call them possible exons. We don't know for sure that they're exons. We want to find out whether they're exons. Okay? We then designed one 60-base probe for each possible exon. And this gives you sort of an overall view of the density of our probes versus the density of exons that are already known. So we're looking at a lot of places that were previously unexplored. I won't go through this part in detail. It doesn't really matter. But our tissue samples consisted of 37 mouse tissues collected into 12 pools. So to keep things simple, you can think of there being 12 tissue pools. And there's an interesting problem of how best to group the data in order to maximize the information or the ability to detect transcripts. And this is now the abstract view of the data set. So if you haven't understood anything talk, that's okay. You can actually just look at this matrix and you can see the problem. So we've got a data matrix. It's got 1 million columns and 12 rows. That's the size of this data matrix. And I've color-coded each measurement using this color scheme here. So white means low expression, so there's a low amount of that transcript detected. Red means there was a lot of that transcript detected. Okay, so again, each row is a tissue, and each column is a different probe, and the probes are arranged in genomic order. Okay, we extracted them from the genome, and I'm showing you those probes in genomic order. So when you look at a sequence of probes here, say in this region, you notice that two or three probes all have high expression in this particular tissue and low expression in the other tissue, the other tissues. And so that means that three positions in the genome that are close together all get activated in the same tissue. And that's evidence that those three locations in the genome are part of the same transcript, right? Because a transcript will involve multiple exons, and all of those exons will therefore be expressed in the same tissue or not expressed. Okay, so groups that look like this are evidence of a transcript. Here's another one over here. So these all have high expression in this tissue pool, low expression in the other ones. When you see a set of probes that are interrupted by a subset of probes that are noisy, that don't belong to the pattern, that's evidence that that extra set of probes just doesn't belong in the transcript. So really, when you look at the data this way, the code that we're trying to detect is really a vector repetition code with deletions. So each symbol, if you like, is a 12-dimensional vector. And what makes this a little more interesting is that it's not actually discrete, because the transcript abundance or the transcript concentration is real values. Okay, so really it's a continuous vector code, and there are deletions just corresponding to these probes that don't correspond to bona fide exons. So let's take a closer look at this, at a particular segment here, and I'm going to tell you how we think of a transcript and how we're going to model it. So we're going to identify a transcript since we don't know the code beforehand, right? Unlike coding, where you know the code beforehand and you just want to detect the code word, we don't know what the code is. So what we're going to do for decoding is actually come up with a pointer into the data set that says this particular measurement is representative of that repetition code. Okay, so here we've chosen this particular probe and said that this 12-dimensional vector is representative of this repetition code, the code word, sorry, in this repetition code. All right, the other information we need to know is the number of probes before this prototype, the number of probes after the prototype. All right, we need to know the boundaries, of course, of the transcript. And then, since not all of the probes in this segment of our experiment correspond to the transcript, we also need binary flags for each probe saying whether it's an exon or whether it's not an exon. So this particular probe clearly doesn't belong with this transcript, and so the flag is turned off. This is not part of the transcript. So that's our model for a transcript. Now, of course, if you want to go through the entire data set of 1 million probes and identify transcripts, you need to deal with competition between different transcripts. So maybe there's a repetition here that seems like it's evidence of a transcript, but there's another transcript that seems to overlap with that one, and the two transcripts really need to compete over claiming probes as belonging to the transcript. So there's a competitive process, and so what we'd really like to do is just set up the analysis in one big model, one big system, just like decoding something like a convolutional code that works its way along the data and outputs these transcripts. So that's what we came up with. So here's a factor graph for this system, and it contains variables shown in purple, which are these actual measurements. So for example, x4 is a 12-dimensional vector corresponding to the expression values in the 12 tissue pools. E4 is this binary variable saying whether or not x4 actually belongs in a transcript. If it does belong in a transcript, then there needs to be some prototype for that transcript. The variable r is a pointer into the data identifying the prototype, and it's a relative index. So if r is +2, that means the prototype for the transcript in which to which x4 belongs can be found 1, 2 positions forward in x6. Okay, so that's how we encode the prototype for the transcript. That's an efficient way to encode it because we know that the prototype for a particular probe is not going to be on the other end of the genome. The other variables, well, t is just a variable that's accounting for the transcript state. So t can be in between transcript, within transcript. It's the end of a transcript, beginning of a transcript. And so as you can imagine, t, the transition between these r variables depends on t. So this function node here, for example, tells you that if you're within a transcript and r was 2, r4 is 2, then r5 will be 1. Because if you take one step forward in the genome, then the relative position of the prototype has just become closer. It's decremented by 1. How about this function node here? Well, this function node here says that if x4 is in the transcript, so this flag is set, okay, and r is, say, +2, that means x4 should be similar to x6. So this function node here just measures the cost of how different x4 is from x6. Okay, and I won't go into a lot of details, but I just wanted to give you a flavor of the structure of the factor graph and how we're trying to solve this decoding problem. All right, well, it looks like there's a lot of cycles, so how are we going to decode the transcriptome? Well, the x's are observed, so we can remove them from the factor graph, and we obtain this factor graph, which is a tree. And so, in fact, we can do exact inference and learning just using the max product algorithm. So we just send messages across the graph back and forth, and we do an exact analysis of the transcriptome in terms of this cost function I just told you about. So here's a summary of the results. First of all, our technique is about 10 times more sensitive than other methods. We detected about 155,000 exons. We predicted about 30,000 new exons. And also, I can't tell you about the details, but we've also reconciled a lot of discrepancies in existing libraries. So these libraries of transcripts I told you about, if you compare them, there seem to be discrepancies, but we were able to reconcile a lot of those discrepancies. Well, what about this overall view, the sort of crude view? What's the number of genes? All right, so let's revisit that. This is the view I just told you about. Well, surprisingly, we found that even though we detected a huge number of new transcripts, all of the transcripts we detected are associated with known genes. So the splicing isoforms or extensions, or just there were errors in the known genes, okay? But the actual number of new transcripts that are isolated in the middle of genome and weren't known before, we estimate as being 0. So John mentioned contentious results. Around the same time as we published our paper, this Phantom Consortium claimed that there were 5,000 new genes that hadn't been discovered, and they had found them. So this is a quote from their paper. And so we wondered, of course, since we just found out that there were none, based on our analysis, we wondered, are their genes really new? So we actually downloaded their data and said, for each of their supposedly new transcripts, asked the question, is it really new? And we actually found that most of them weren't. So this appeared in Science in March 2006. We actually found that about 3,000 of their proteins, in fact, are splicing isoforms, or these alternatively spliced versions of the same gene. All right, so the Phantom Consortium revised its number from 5,000 to 2,000. That's not the end of the story. We actually had the last word. We actually did a careful false positive analysis, false detection analysis of their method, and estimated that, in fact, about 2,000 of their detections are expected to be false detections. And so our conclusion is that really there are maybe only a few hundred new genes in their set. All right, so let me finish up with a few remarks that I think will be relevant to sort of gluing you together with sort of these interesting problems in molecular biology. So open problems, of course, if you're not an expert in this area, it's hard for you to really know what the problem is. But some comments maybe are helpful. We still need to complete these libraries. So the community has pretty much converged on the library of protein coding genes for humans. And specifically, I think we've converged. But there's a huge number of alternatively spliced transcripts that need to be investigated. And as I said, I think there are codes and signals that can be used to detect these transcripts. And also transcripts that don't make proteins. So I told you about these non-traditional transcripts that actually are playing an increasingly important role in our view of how the transcriptome works. Also, understanding the functions of transcripts. So we've got a library, but what do these things do? How do they interact? What are their functions? I also told you about transcription and splicing and how these processes are regulated by the existence of code words and signals. Well, we still don't really understand the mechanisms. We've just really begun to figure out what's going on there. We don't have a good comprehensive view of what's going on or what are all the code words. And also, there's another topic, and I don't really know what this means. There's an area called genetic networks of genes. You've probably seen pictures where there's a node for each gene and there's edges connecting them. So it looks cool. Maybe there's some potential for research there. So should you work in computational biology? Well, the pluses are, and this is a question I asked myself 5 years ago when I considered entering this field. Well, it's satisfying because it's a major scientific frontier, okay? It's science, not engineering, at least at this point in time. So if you're interested in science, it's exciting. And also potential for high impact on society. So as an engineer, that's also appealing. High impact on society. Now, the minuses, really, it's if you look at biology, and I certainly would have said this 10 years ago, it's mostly a collection of facts, and that's a bit tiresome. The mechanisms are complex and beyond our control. We can't engineer them. They're nature. And also, molecular biology is lacking a mathematical framework. So unlike physics, where there's a mathematical framework, we don't have that in biology. However, but you can also think back to the days, the beginning of communication theory. You know, it also lacked a mathematical framework. So we shouldn't be, thinking really of that as a minus. In fact, you should think of that as a plus, right? If you could be the one to introduce a mathematical framework in molecular biology, think about the importance of that contribution. Just a few comments. How do you enter this field? And this is very important. From my point of view as a professor, hire a tutor, which means a student or a postdoc. This is a person from whom you can extract information. You can ask them dumb questions, and they have to answer them. Also, you'll have to hire a programmer because there's a lot of nitty-gritty into taking the genome and breaking it into pieces and finding sequences and things like that. There's a lot of scripts that need to be written. Pick winner projects, right? So meet with biologists, figure out, is this a winner project or a loser project? And even if it seems to be a winner project, it could develop into a loser project. And then the best thing to do is drop it, right? So I was involved in a project, lasted for about 2 years, spent probably a couple hours a week on it, and then just dropped it. Cold turkey, nothing came from it. But you have to make those decisions and be careful to make them properly. And mutually beneficial collaborations are important. The reason for this is that you have to build trust with a biologist. I see this happening, and it's hilarious. The information processing researcher will come to the biologist and basically say, give me all your data. I want to get recognition for discovering things in it. And that's of course the wrong attitude. You need to approach the biologist and say, I'd like to start working with you from square one. Let's design these microarrays together. How are we going to reduce the noise? How are we going to produce better data for you? If you work with them from step one, then you gain a level of trust, which means you're really authentic collaborators, you're not just trying to take their data. Okay. How long will it take? Well, but roughly that long. So it takes about well, it depends, of course. If you hire your tutor, you got to hire a tutor. If you're a postdoc, this is risky, switching into the field, because you'd like to get results in a short period of time, like 2 years. I think that's going to be tough. If you're a student, you can probably learn about it in 4 years. But if you have the right support, so if you're a principal investigator and you have a tutor and a programmer and so on, it takes about 4 years to get up and running in this field. So it doesn't actually take that long. Of course, you should make sure to pick winner biologists to collaborate with, people who know what they're doing and know what the interesting questions are. All right, so I'll leave it at that. You can take a look at this website. On Friday, I'll have posted information. I'll posted this set of slides, and I'll also have posted pointers to helpful papers and databases and things like that. And I'll just finish up by acknowledging contributions from Quade Morris. So Quade Morris was my tutor, and he did very well in this area, and he's now a faculty member at the University of Toronto in medicine. Leo Lee, he's the one who did the work on this that appeared in Science, where we analyzed the results of the Phantom Consortium, and my students, and also my collaborators, Tim Hughes, working on microarrays, Ben Blenko, working on alternative splicing, Andrew Emily, who does proteomics, Charlie Boone, who does genetic networks. Thank you.
We have time for a few questions or comments. Here.
So there's one question about why, which will move us from collection of facts to a little bit of science. So we know that encodings are very well preserved. And the question is why. Is it possible that the introns have some redundant bits that are used to correct errors whenever they occur in exon, or you can only explain them through Darwin's theory?
So the question is, exons are conserved across different species? That's first of all the observation. Sorry?
Even within the one DNA, there are certain parts that are much better preserved than the others. The question is why.
So I guess the question is just generally in the DNA, why are certain regions conserved more than others? And that's an interesting question. Really, there's many different competing problems. One problem is that the DNA would like to be able to introduce diversity, because that's how evolution occurs. Of course, if there's too much diversity, then the organism will degrade. And so there needs to be a balance of these two effects. Not only that, but you can see there's different levels of what the sequence data is doing. There's the level of what's going to go into the protein. There's the level of what's going to go into a transcript. But then there's also these regulatory code words. So you can't just, if you just take away part of the DNA that appears to do nothing and use it for making a protein, you may have stolen away a code word that's important for regulation.
Another question.
Yeah. So the question is, so we had the last word, right? So did they concede? Actually, I don't think they understood our point about the false detection rate. Yeah. So their response indicated that they didn't really understand that we had analyzed the number of false detections. Yeah.
Shlomo, yeah.
Is the question, what role does evolution play in our...
Can they having some kind of an evolution signature, meaning that with evolution, introns change in the way that we cannot read it now. Once maybe we will. And then we will know this is the DNA of down the road of the evolution. This is just some kind of time. Evolution does not necessarily go with time, but some kind of encrypted evolution signature.
And so the point, the observation is that evolution may be there may be a signature, in a sense, of evolution in our current DNA. Yes, that's true. Is there a question or an observation that follows that? Okay. Yeah. Yes, there is.
One more in brown.
With respect to the genome, that defines some set of features, and I presume that the transcription set is based on that set. My question is, what happens outside that set? And is that transcription invariant, or how does it change?
So certain code words or certain features in the genome activate transcription. And you're asking the question, is sort of outside of those regions, outside of that kind of control, what's going on? Right, so I guess there are two answers. One is, of course, we don't know what all the regulatory elements are, and so there could be transcription in regions for reasons we're just not familiar with. But then really anything goes. That's also exciting about, something that's exciting about this field, is that there used to be the central dogma of how molecular biology works. And when someone familiar with information processing looked at the problem, it was just mind-boggling to remember all these facts in the central dogma. But increasingly, biologists are discovering that all that old stuff is just wrong. So I think a good way to approach the problem is not to be too restrictive. You do need to learn some of the terminology and sort of the current view, but not to be too restrictive, and really just to think of it as an open problem. Essentially, there's a genome. There's many ways of activating the genome. We don't know all of them. Anything's really possible. Yeah.
I think we have to stop the questions. Brendan will be here today, I think, at least today. And it is difficult to discuss in here, so we can